In this class, I am explaining about gene regulation in prokaryotes. In this syllabus, we discussed about introduction to gene regulation in prokaryotes, types of operon in gene regulation and mechanism. First one, introduction to gene regulation in prokaryotes. Gene regulation refers to the control of the rate or manner in which a gene is expressed. Gene regulation is the process by which the cell determines when and where genes will be activated and how much gene product will be produced. Thus, the gene expression is controlled by a complex of numerous regulatory genes and regulatory proteins. The gene regulation has been studied in both prokaryotes and eukaryotes. In prokaryotes, the operon model is gene regulation is widely accepted, and it is developed by in prokaryotes. The operon model of gene regulation was developed by Jacob and Monod in 1961, for which they were awarded Nobel Prize in 1965. Now this model of gene regulation is widely accepted and organism used. The operon model was developed working with lactose region, that is lac region of human intestine bacteria, that is Escherichia coli. The gene regulation was studied for degradation of the sugar lactose and gene involved. In the operon model of gene regulation, four types of genes are used. First one, structural genes, second one, operator gene, third one, promoter gene, and the fourth one, regulator genes are involved. And the main enzymes involved in lac operon, four types of enzymes are involved in gene regulation of prokaryotes. These are beta galactosidase and galactosidase permease, transacetylase and RNA polymerase. The beta galactosidase catalyzes the breakdown of lactose into glucose and galactose. The galactosidase permease permits entry of lactose from the medium into the bacterial cell. The enzyme transacetylase transfers an acetyl group from acetyl coenzyme A to beta galactosidase. The enzyme mRNA polymerase controls on-off of the transcription. And the types of operon in gene regulation. In prokaryotes, operon are of two types, inducible and repressible. The example of an inducible operon is lactose operon, which contains genes that encode enzymes responsible for lactose metabolism. An example for repressible operon is the trip operon, which encodes enzyme responsible for the synthesis of the amino acid tryptophan. And first one, inducible operon. Inducible enzymes. An enzyme whose production is enhanced by addition the substrate in the culture medium is called inducible enzyme, and the system is called inducible system. The example of the inducible operon is the lactose operon, which contains genes that encode enzymes responsible for lactose metabolism. In bacteria, operon refers to a group of closely linked genes which act together and code for the various enzyme of a particular biochemical pathway. This is the structure, the model of log operon of Escherichia coli, this like. And the structural genes. There are three structural genes of LAC operon, that is LAC-Z, LAC-Y and LAC-A. The main function of structural genes is to control of protein synthesis through messenger RNA. Function of these genes are, first one, LAC-Z. LAC-Z, it encodes the enzyme beta galactosidase which catalyzes the breakdown of lactose into glucose and galactose. And LAC-Y, it encodes the enzyme galactosidase permease which permits entry of lactose from the medium into the bacterial cell. And LAC-A, it encodes the enzymes transacetylase which transfer an acetyl group from acetyl coenzyme A to beta galactosidase. And next gene is promoter gene. The three structural genes are under the control of promoter gene. In the promoter, RNA polymerase binds to the DNA and prepares to initiate the transcription. The main function of promoter gene is to initiate mRNA's transcription. And the next gene is operator gene. The other regulatory element in an operon is the operator. This is the element that determines whether or not the genes of the operon are transcribed. The main function of the operator gene is to control function of structural gene. And next one is regulator gene. The regulator gene expressed all time or constitutively and plays an important role in operon function. This is the LAC1 gene which encodes a protein called LAC repressor. The LAC repressor has two functional domains or regions, one that binds to the DNA of the operator region and that binds to a lactose. This is the LAC operon. And when repressor binds to operator, it prevents RNA polymerase advancing along the operon and transcription does not occur. The regulation of the operon depends on regulating whether or not the repressor binds to the operator. The function of regulator gene is to direct synthesis of repressor, a protein molecule. Its function differs in the presence and absence of lactose. And when lactose is absent, in the environment, events takes place in this way. The LAC1 gene is transcribed and the mRNA is translated, producing the LAC repressor. The repressor binds to the operator and blocks RNA polymerase. When RNA polymerase is blocked, there is no transcription. Thus, the enzyme for lactose metabolism are not synthesized because there is no lactose to metabolize. Thus, when lactose is absent, lactose metabolized enzyme are not produced. And when lactose is present, in the environment, the events occurs in a different way. A small amount of lactose enters into the cell and affects regulation of the operon. The LAC repressor is still synthesized, the repressor can bind to lactose. After binding to lactose, the repressor undergoes a conformational change, and the molecules that change shape when they bind to another molecule are called allosteric molecule. With this change, the lac repressor is unable to bind to the operator region. Hence, RNA polymerase is not blocked and is able to transcribe the gene of operon. The enzyme encoded by those genes are produced. The lac permease transports more lactose into the cell, and beta galactoside cleaves the lactose into glucose and galactose. This can be further metabolized by other enzymes producing energy for the cell. Lactose therefore is able to induce the synthesis of the enzyme necessary for its metabolism. Lactose is the inducer of lac operon. Thus, when lactose is absent, lactose metabolizing enzymes are not produced. When lactose is present, those enzymes are produced. And merits of operon model in gene regulation. It is very simple yet informative model to gene regulation in prokaryotes. It is very well understood model of gene regulation in prokaryotes. And third one, this model is based on empirical results and has been studied on different prokaryotes. Thank you.